Good evening, everybody. My name is Shane Holmes. I live in Long Lake in the Central Adirondack Park. I have an undergraduate degree in biology from St. Lawrence University and a Master of Science in Information Technology from Purdue. I've been a part of the Master Naturalist program now for about a year. This is my second presentation for the Naturalist Exchange. I chose to give this talk because I'm very interested in how DNA is used in conservation. My interest began back in 1993 when I did my senior honors thesis on the analysis of mitochondrial DNA of the eastern coyote. This year, I'm hoping to work with a nonprofit citizen science group studying brook trout genetics in the Adirondacks. But tonight's presentation is about understanding environmental DNA and its impact on conservation. We're not going to go too deep into the technical side of genetics and the tools for analyzing genetics, but we'll cover the basics and help you understand why it's important in the study for conservation. And if we have time, we'll go over some real world examples. So you can think of it a bit like being nature detectives. Instead of looking for criminals, you're looking for secrets about the environment. So imagine that forests or lakes or rivers or even the air could hold clues about the organisms living there. But these clues are hidden, kind of like invisible ink. Scientists use spectral technology to find and read these hidden messages, helping them to learn more about how to protect our natural surroundings. Kind of like in the same way that forensic scientists have worked out how to decipher fingerprints left behind everywhere we go. Fragments of eDNA or environmental DNA can tell a great deal about who left those invisible traces. And each species, in fact, has its own DNA sequence, kind of like a genetic barcode. So what exactly is environmental DNA? Well, it's the genetic material left behind by all living organisms. It's shed into the environment by their skin, feces, or even mucus. You know, it could be like a strand of fur caught on a tree branch, a fragment of skin drifting through the water, or even ancient bones buried in sediment. Each of these helps explain the piece of the ecosystem story. And environmental DNA is very important because it lets us know what organisms are around and what they were doing without actually seeing them or catching them. So when we talk about environmental DNA, and we mean the genetic material found in every cell of every living thing, we're talking about the DNA, the molecule shared by all life forms. This is animals, plants, fungi, bacteria, and it's unique to each individual. In animals, environmental DNA usually refers to two main types of DNA, the nuclear DNA and mitochondrial DNA. For plants and organisms like algae, it's the chloroplast DNA. In studies involving things like microbes or other forms, DNA is plasmid DNA and viral RNA. That's what might be used. But we're not going to cover these today. We're going to focus on the three most commonly studied types when we're dealing with environmental DNA. That's nuclear DNA, mitochondrial DNA, and chloroplast. And to understand environmental DNA, it's helpful to know how these types of DNA differ. So we're going to go back a little bit to our early biology days and talk about things like nuclear DNA, which is found in the nucleus of almost every cell in the body. And it acts as a blueprint for growth and development. It comes from both of the parents, half from your mom and half from your dad. And it determines traits like hair and eye color. This unique mix makes each person different, except for identical twins. Although it contains a lot of detailed information, it's less common than the other types of DNA, like the mitochondrial and chloroplast. It breaks down faster because there's less of it, and it's in larger pieces. It can be more easily damaged by things like sunlight, heat, and bacteria. This makes nuclear DNA harder to use for detecting species in degraded samples like in water or environments where DNA is scarce. Then we have mitochondrial DNA, which is a special type of DNA found in the mitochondria, which are parts of the cells that produce energy. Unlike the nuclear DNA, mitochondrial DNA is passed down only from the mother, making it easy to trace maternal ancestry. It's more common, it's smaller, it has a circular shape, which makes it more stable and less likely to break down. Because of these features, mitochondrial DNA is easier to find, even in damaged samples. And since it changes very little over time and it's highly abundant, it's often used when studying environmental DNA, especially when working with scarce or degraded DNA like what's found in water. Then we have chloroplast DNA, which is found in the chloroplasts of plant cells and certain algae. And it's different from the nuclear DNA. It usually comes from the mother, like the mitochondria, but in some pieces, it can be inherited from both of the parents. Like mitochondria DNA, chloroplast DNA is found in many copies within the cells, making it more stable and less likely to break down compared to nuclear DNA. However, environments like where there's lots of microbes or harsh conditions, it can break down quickly. Because it's more abundant and it breaks down slower though, chloroplast DNA is useful for tracking plant and algae species, especially in places where the DNA normally degrades quickly, like in water. So when we look at this genetic material, we have to look at how it plays a role in conservation. These 4 items listed here are just a small subset of the ways environmental DNA is being used in research projects related to conservation around the globe. We'll take a look at these individually. Biodiversity assessments. Environmental DNA can be used to assess the biodiversity by collecting samples from within the environment, allowing searchers to identify a wide range of species present in an area without the need of direct observation or capture. Environmental DNA can also help in the early detection of invasive species by identifying their genetic material in the environment, enabling quicker management actions to prevent spread of the species. Environmental DNA can also facilitate in the tracking of endangered species by detecting their presence through minimal non-invasive sampling, thus aiding in monitoring their populations and habitat usage without actually disturbing them. And then in aquatic environments, environmental DNA analysis provides insights into the species composition and health of water bodies, helping the research understand economic or ecological dynamics and impacts of human activities on these ecosystems. And like anything else, there are limitations. Environmental DNA is a valuable tool for detecting species, but it degrades quickly due to factors like temperature, UV light, and microbial activities. This creates a narrow window for detection, making timely sample collection crucial. This also makes it tough to determine how recently an organism was present. Since environmental DNA can persist in the environment for varying lengths of time, it's challenging to pinpoint the exact timeline of its presence. This can lead to uncertainty in assessing current populations versus historical presence. Also, the risk of cross-contamination can lead to false positives. This occurs when DNA from an organism is inadvertently introduced into a sample from external sources, such as equipment, researchers, or even a nearby environment. Even tiny amounts of contaminated DNA can skew results in falsely indicating the presence of species that weren't actually there. So when researchers and scientists are looking at environmental DNA, what are the actual steps? Well, the steps begins with the process of sampling, where the environmental samples taken from water, soil, or air are collected to capture DNA shedding by organisms. Then this is followed by an extraction process where the DNA is isolated from the sample. And then next, the DNA undergoes an amplification using techniques such as polymerase chain reaction to increase the quality of specific genetic markers. After it's been amplified, sequencing is used to identify the genetic sequences present, allowing scientists to determine the species that contributed the DNA. The analysis phase involves bioinformatics tools to compare these sequences against databases and interpret the findings. And finally, the results reveal the presence or the absence of species in the environment, offering insights into biodiversity, the health of the ecosystem, or the effects of conservation efforts. Let's look at these a little bit closer. Environmental DNA can be sampled from the environment using a variety of methods, depending on the habitat and the target species. In terrestrial environments, environmental DNA is often collected from soil, where organisms leave the DNA through waste or skin cells or decaying material. In water, you can use water filtration, where the water is passed through the filter to capture DNA particles, ideal for monitoring species in rivers, lakes, and oceans. And sediment sampling from the bottom of these bodies of water can provide historical data on species presence by extracting the DNA from its layers. And recent advances allow for air sampling, where filters can capture airborne DNA from organisms such as insects or pollen. In colder climates, snow and ice can be collected and melted to extract environmental DNA. This can provide insights into the presence of species in polar and alpine ecosystems. These methods offer the researchers flexible, non-invasive ways to monitor biodiversity across diverse habitats. And a lot of these are done simultaneously. And what I mean by that is they might do water samples, soil samples, and air samples in the same day. In the eDNA extraction process, the DNA is isolated from the sample. The collected sample is treated with a series of chemical reagents that break down cell membranes and proteins and release in the DNA. The DNA is then separated from the other materials, such as sediments or organic debris, often through centrifugation or filtration. The extracted DNA is purified and concentrated, ready for subsequent amplification and sequencing to identify the organisms present in the sample. This step is critical for obtaining clean, usable genetic material for further analysis. And the amplification process is where the environmental DNA analysis involves increasing the quantity of a specific DNA fragment to make it more detectable for further study. This is typically done using a method called polymerase chain reaction, which targets and replicates specific genetic markers associated with the species of interest. By creating millions of copies of these DNA sequences, even smaller degraded samples can be analyzed. In conservation, amplification is crucial for detecting the presence of rare or elusive species, as well as monitoring biodiversity in ecosystems. Now, PCR has been around a long time. It was available back in 1993 when I did my analysis of mitochondrial DNA on the eastern coyote, and it's still used today. However, there are many more high-tech techniques. I brought up PCR because it's one of the more well-known ones. The sequencing process in environmental DNA analysis involves determining the precise order of nucleotides, or the building blocks of the DNA, in the amplified fragment. After it's been amplified, the DNA is run through a sequencing machine, which reads the genetic code of the sample. This sequence is then compared to reference databases that contain the known sequences of various species. In conservation, sequencing is vital for accurately identifying the species present in a sample. There are many databases available today. Gen Data, I believe, is the name of one that people use. The analysis process of environmental DNA involves interpreting the sequenced DNA data to identify species and assess the biodiversity in a given environment. After sequencing using bioinformatic tools to compare the DNA, the sequences, again, are referenced to databases such as GenBank that contain genetic information from all known species. This comparison allows scientists to match the DNA sequences to specific organisms, determining which species are present in the sample. In conservation, this step is crucial for identifying rare, endangered, or even invasive species, also for monitoring ecosystem health and tracking changes in the biodiversity over time. This analysis can also provide insights into the population dynamics and species interactions, as well as helping conservationists make data-driven decisions for managing and protecting ecosystems in the future. So in the end, we have results, and in conservation, these results can reveal the presence of specific target species, such as endangered animals, invasive species, or key indicator species that reflect ecosystem health. Results offer a snapshot of the biological community in a given area, helping conservationists understand species distribution, track changes over time, and assess the effectiveness of conservation actions. This data can be used to guide management decisions such as habitat restoration, species protection, and environmental monitoring, making it a very powerful tool for shaping conservation strategies. So I have some real-world examples of environmental DNA conservation here that I want to talk about and the role that they play in conservation. Again, I don't have a lot of time to cover the technical aspects of each of these, so I'm just briefly going to cover a little bit about each. However, I do provide references at the end for each of these. Tracking marine biodiversity and coral reef ecosystems. This study demonstrates how environmental DNA is applied to monitor and assess the biodiversity within a coral reef ecosystem, which are among the diverse and threatened ecosystems on our planet. The case focuses on how environmental DNA sampling compared to traditional survey methods provides a non-invasive, efficient way to track marine species and to detect even cryptic or elusive organisms that might be missed by visual surveys. This method has proved crucial in understanding species composition, ecosystem health, and the effects of climate change or coral reefs. This study often highlights how environmental DNA allows for more comprehensive biodiversity assessments, supporting conservation efforts by helping identify priority areas for protection and restoration. So imagine just by dipping a container into the water, taking a sample of water, or maybe running a sieve net through the water and collecting samples, how you can do a study without interfering with any of the coral reef. Another one is the detection of invasive species. And in this case, the detection of invasive Asian carp in US waterways. This case study highlighted how environmental DNA was used as a powerful tool to detect the presence of Asian carp, a very highly invasive species that threatens the Great Lakes ecosystem. Traditional survey methods like netting and electrofishing were very labor intensive and often missed the early stages of an invasion. In contrast, the environmental DNA allows researchers to detect the presence of the carp from just water samples when the population density is extremely low, giving the conservationists and policymakers a head start in making preventive measures. The detection of environmental DNA from Asian carp in waterways near the Great Lakes serves as a critical early warning system, prompting increased efforts to prevent the spread of this species, which can significantly disrupt native fish populations. And then another one is the endangered species of, in this case, this is a Rocky Mountain frog and a giant Iowa salamander. Monitoring endangered amphibians in remote areas was this study, and the study exemplified how environmental DNA was employed to track endangered amphibians in habitats that are otherwise difficult to access or survey using traditional methods. In this case, environmental DNA was used to detect the presence of endangered amphibians, such as this certain frog or salamander, in remote and often inaccessible environments like wetlands or mountain streams. The non-invasive nature of environmental DNA allowed researchers to gather data on species distribution and population status without the need for physical capture or visual confirmation, which can be particularly challenging for amphibians that live in hard-to-reach areas. This method also minimized habitat disturbance, which is crucial when working with endangered species. This study showcased environmental DNA as an efficient and reliable way to monitor amphibian populations, contributing to a more informed conservation decisions and helping prioritize areas for protection and recovery efforts. So those were just 3 examples out of many examples out there. And I'm going to talk about another one in a bit, but first, I want to talk about the future of environmental DNA and conservation being shaped by things like advances in next generation sequencing, as well as its integration with artificial intelligence and machine learning, which will enable faster and more accurate analysis of complex genetic data. These technologies can rapidly process vast amounts of environmental DNA sequences, identifying species more efficiently while reducing human error. And artificial intelligence-driven algorithms can help detect patterns and trends in biodiversity over time, helping scientists better understand ecosystems and predict changes. This integration enhances the scalability of environmental DNA research, making it more of a powerful tool for large-scale conservation efforts and environmental monitoring. Everybody knows about the fast overtake with artificial intelligence today. You know, we know about ChatGPT and all the other fancy things that are coming out. A lot of this is being applied also in conservation efforts as well. So the other one I wanted to talk about was using these advances in environmental DNA technology. They're hoping to improve and understand our ecosystems worldwide, but they're also helping with local conservation efforts. Particularly here in the Adirondacks, researchers are using these methods to study things like brook trout by collecting their genetic samples to map populations and assess factors like stocking and environmental changes. This research aims to improve the health of these fish, while another study focuses on refining environmental DNA techniques to monitor these fish in remote streams. And researchers are always trying to improve their ways to monitor these brook trout in hard to reach streams. They're finding better methods to collect water, like optimizing the amounts of water taking and using special filters to capture the environmental DNA more efficiently. They're also enhancing lab techniques to allow them to detect smaller amounts of environmental DNA, making it easier to find the brook trout in even some of the tougher conditions. By using advanced data analysis, they can accurately identify different species and assess genetic diversity. And then by standardizing how the samples are collected and analyzed, they can ensure consistent results across studies, which is essential for long-term monitoring. And then combining their findings with the environmental factors like water temperature, it can help researchers understand what affects brook trout populations and how it shapes conservation efforts. And overall, these improvements enable more effective monitoring and better conservation strategies for the brook trout. particularly in challenging areas. And that is the end. That was rather quick. I think I talked too fast, but I'm going to open it up for questions and discussion if anybody has any.
Thank you, Shane. There are some questions coming in. I have one. I'm curious because you've worked with brook trout. Since brook trout are often in like small headwater streams, cold water streams, and they can be kind of flashy, right? Like high water flows and then low water flows. Do you have a sense for what distance away from the trout's location that DNA can be detected at?
Yeah, so it has been detected great distances, particularly in moving water, because the DNA, of course, flows in the water. And even in lakes and ponds, or even some of the larger lakes, turbidity, the movement of water based on wind and stuff also helps the DNA debris to move through not just across the water, but through the layers of the water as well.
Yeah, I can imagine that knowing that would be kind of important for tying it to, habitat conditions or changes even over time, and knowing, is that trout there or is it, how far away is it, is it? Okay, well, I have some questions here. Steve Jacoby asks, of all living organisms, what percentage have had their DNA sequenced so that it can be used for ID?
I believe all known North American species, particularly mammals, have been sequenced. And I'm pretty sure a very large amount of plants, fungi, and algae, have been sequenced as well. Of course, there are many, many, many species that have yet to have been identified. And we're still learning of those species. And I'm sure many, many times through the study of environmental DNA, some new species are found as well.
You have a sense for insects?
Even for insects as well. Yes. Yeah, insects.
Okay, Adrian asks, how did they identify species initially, and what precautions do collectors take to avoid contamination of samples?
How did they identify species initially? Yeah, so, most of us know that in the past, manual research was done using traps and, things like game cameras and visual identification, lots of slow, labor-intensive ways of trying to identify species. And what precautions do collectors take to avoid contamination of samples? Well, they are really learning as they go on this. Contamination of samples is a big part of the analysis process and a big issue, depending on how you take the sample, would be how you worry about how you might contaminate it. So for instance, taking a sample with a net, you would hold the net by a handle and move it through the water, pulling the net out of the water without actually touching the net with your hands. And then, emptying the net into a container without actually physically touching the net. You could also use things like gloves, to try and prevent any contamination of samples. But the big thing here is I think what you're getting at is how do you eliminate the contamination by things like air? I think that's what you're getting at, right? So if I'm taking a water sample using a net and I'm pulling out of the water and then trying to get it into a container, how do you prevent the air from also contaminating it? And that, I think, is probably impossible. So they're trying to learn how to analyze the sample taken from water that might also contain contamination from like the air as well. I don't know if I answered your question or not.
I think Alan's question, approximately what % of species have been sequenced, was answered previously. Probably difficult to know what % of species, since we still don't really know how many species we have, but.
Yeah, they do have a very large amount of species sequenced. There's no way they have them all. I mean, yeah, not every insect, not every plant, not every fungi has been identified.
You asked, please speak to the challenges of sortum. Is that sorting?
Yeah, can you hear me okay?
Yeah.
Yep.
Yeah, I accidentally hit enter before I finished the question, but the bottom line was, and I confess in ignorance, if I'm understanding you correctly, you can collect DNA from of an organism just from the water the organism has been in. Is that correct?
That's correct.
Okay, so if you're out there trying to see if a particular species is there and you're using that method to collect the DNA samples, I'm assuming there's a lot of different samples of different organisms in the water at the same time. And is it challenging to find the one you're really looking for? Well, first of all, is that a problem? Do you inadvertently collect a lot of samples of a lot of different organisms, given the fact that it can be, I guess, dissolved in the water? And is it a challenge to sort through and find the one you're actually looking for?
It is a challenge, and a water sample holds a lot of different type of species across, all the different types of kingdoms. You're talking about cells that could potentially come from fish, cells that could potentially come from, zooplankton, cells that could come from plant material. So, lots of different species. So they have ways of extracting. We didn't talk about any of those techniques, but they have ways of extracting specific DNA types, whether it's nuclear DNA or mitochondrial DNA. So if you're eliminating animal species from plant species, of course, you're extracting either mitochondrial DNA or nuclear DNA. Whereas if you're looking specifically for a plant species, you might want to eliminate mitochondrial and nuclear DNA by just going after the chloroplast DNA, if that makes any sense.
Yeah, definitely. Man, it's amazing that all that stuff can be in water. My goodness.
Yeah. And and they're learning new techniques every day. I talked about refining these techniques, and they're doing studies specifically to refine techniques. So they're not so much worried about the results at the end in terms of how it might benefit conservation efforts right away, but more about the techniques using to identify and sample and do analysis on the results. So they're doing studies to do that in particular.
Fascinating. Thank you.
Refine the techniques. Yep. And I know the brook trout study that I want to get involved with this spring, I believe it actually is with Cornell as well, Christy. It's with a citizens science organization called Trout Power as well with Cornell. I believe they've collaborated together to do a study of brook trout to refine the techniques for studying the brook trout environmental DNA.
Interesting. Do you know who's doing that at Cornell? Do you think it's
From
Pete
Cornell?
Mackin?
No, I don't. I didn't get any names.
And maybe somebody in my department even. I'd be curious if they're looking for other volunteers. I'd be interested to know who the contact is, because there may be other master naturalist volunteers across
I
the
do
state
have the
would be-
contact at Trout Power, and I can find out who it is at Cornell that they're working with.
Okay, that would be excellent. Thank you. Paul Siskin asks, when a researcher wants to use eDNA data, after they collect the samples, do they generally process the DNA themselves, or do they ship the samples off to another lab for processing?
Aha, that's a really good question. Yeah, so both. For the most part, pretty much not done in the field. It's done in a laboratory. It was back in 1993 when I collected liver samples from coyotes. You take it, in this case, when I was studying coyotes, it was a liver sample that was put in ethanol and was stored in little containers in ethanol. But yeah, it's taken back to a laboratory. It's either done by themselves in a laboratory, a laboratory, for instance, that Cornell has, or they do, and they can ship it off to for-profit laboratories that actually do this type of stuff. Yeah, which is done quite frequently. Because these for-profit companies have really refined these techniques.
Thank you. Catherine asks, will it be possible to identify areas where the jumping worm and hammerhead worm are in the soil so as to try to remove them?
I've never even heard of either of those species. So what was the question again?
Would it be possible to identify areas where basically worms are in the soil, various species of worms?
Yeah, worms shed lots of mucus. So I think that would be a very easy species to study based, using soil samples because they excrete so much mucus.
Okay. Dave and Ann ask, in your analyses, how accurately can you determine the location of an organism given that the DNA samples may have traveled some distance? So it's similar to my question.
How accurately are you able to determine a species presence?
Well, the location of an organism, given that the DNA samples could have traveled some distance after being
Yeah,
shed.
that is very tough. So, when we're talking about a body of water, really the only thing you can determine is efficiently 100% that it exists in that body of water and not particularly a specific location. Let's take the St. Lawrence River, for example, which runs from the Great Lakes to the Atlantic Ocean. Taking a sample of, let's say you're looking for the American eel and you're taking a water sample, are you able to determine the exact location of that eel or just the fact that it exists in that river? And I think just the fact that it exists in the river. Now, in the future, will they be able to determine specific locations? Maybe. I'm not sure if they're able to do that at this time. And that also goes with determining how long they've been in that particular environment. So for instance, maybe you're finding some genetic material of the American eel in water samples and in soil samples. Can you determine how long that specific species has existed in the water? A lot of those studies are happening today, particularly here in the Adirondacks, where they're trying to determine what species are indigenous to the Adirondacks. They've fought over for years about things like the yellow perch and whether or not the yellow perch existed in the Adirondack Park back in the day when indigenous fish such as brook trout and lake trout existed and were determined to be the only species of fish that existed here. I hope I answered your question.
Let's see. Diane Francis she has a question about wolves and wolf DNA, and she asked if she can contact you afterward to
Yeah.
do a couple questions.
Yeah.
Can
You
I
can.
give your email in the chat? Is that okay?
Yes, you can.
Okay.
You can give her my email, but just quickly, my study of the mitochondrial DNA of the eastern coyote was to look at whether or not they were actually hybridizing with domestic dogs. There was this theory back when I was growing up of this, coy dog, the existence of a coy dog in the North Country, and this idea that coyotes were breeding with feral, wild, or domestic dogs, and we determined that they actually weren't. So, although we did sequence some wolf genetics, it wasn't particularly looking at wolves. It was looking at domestic dogs.
I'll put your email. Do you know your email off right off? Top of your-
Holmes Shane A at Yahoo.
So, okay, Shane A at Yahoo. Like that?
Oh, let's see, put it in the chat.
Is it cap sensitive?
Holmes Shane A at yahoo.com. Yep.
Okay. All right. Let's see. There are some other questions. How large does a DNA sample need to be to allow a positive identification?
Well, considering the DNA sequence is microscopic, I don't know the exact size that they have to be. Very minute. It does not take a very large sample to get a lot of DNA. And in my case, with a coyote liver sample, it was just a very, very small trim piece, that was smaller than the end of your pinky. And we got just from that one little piece, we could do multiple runs with the DNA, gel runs, and still have lots of material left over. But that's tissue. What you're referring to is, samples collected from like a water sample and things like that. So water holds a lot of material. So, who knows how much in each sample, how much genetic material exists in each sample. And I think it changes, with each sample. So I think it would be tough to determine, an exact quantity of genetic material that you get out of a particular sample.
Tracy Lemonek asks, as an analytical chemist, I see this as a qualitative analysis. How do you make it quantitative? Are you able to determine individuals from the DNA so that you could make some population estimates?
Right, you can. I was trying to think of a better way to answer that, but that's a good way. Yeah, so, if that's exactly what your study is looking for, numbers, or are you looking for just the existence of, for instance, invasive species? You might be just looking whether or not invasive species exist there before you determine the extent of numbers. But in terms of numbers, yeah, that's one of the big areas that they look at is in biodiversity, is how many of a particular species exists. They have specific techniques for determining, equations that I didn't utilize in this presentation, but they have equations for determining specific numbers based off a specific sample of genetic material. And these labs provide that.
David asks, are there other species-specific chemicals or proteins that can be detected? He said, I don't have a specific example in mind, just curious if there are any.
In terms of different from like a DNA nucleotide?
David, do you want to unmute and clarify?
Yeah, just if the sampling techniques, if they're also looking for other like trace chemicals or pheromones or so. I'm just thinking, I know that there are species specific ones. One doesn't come in mind. I was just curious, along with the sampling for DNA, is there other chemical markers that they might be looking for, or biological or just or
Yeah, I'm sure there
in
are.
any sense?
I don't know of any particular study that's doing it in combination. But I'm sure there are. They could, God, trying to think of some proteins that, there are a lot of proteins found in blood, but blood's more of a macro
Yeah,
level.
I was thinking like maybe like pheromones or something. I don't
Yeah,
know.
I don't know of any off the top of my head. Sorry.
Let's see. Thanks for the info, Mandy, about Keith. Yeah, I'll look into that further. All right, before I ask this last question, I just want to say that 2 summers ago, we had our volunteer weekend at Cayuga Lake. We were studying harmful algal blooms. And during that weekend, a graduate student from my department, Alex, he's working with DEC on a Cisco reintroduction project in Cayuga Lake, and he talked about how he's using eDNA to detect whether the Cisco were there, where they were in the lake, and they also then captured and tagged some Cisco and then had a machine that could read the tags when the Cisco passed by. So I think they were doing some
Oh, yeah.
confirmation in terms of how effective the DNA was, the eDNA was, and if it could be confirmed. But there are a number of people. I'll also
Can I say something really quick, Chrissy?
sure.
That's a great example of what I was talking about earlier about refining techniques. And in this case, they're using a macro level, the tagging of the fish, and a micro level, which is the study of the environmental DNA simultaneously to determine accuracy in one or the other, right?
Mhm.
Yeah, great example.
Yeah, absolutely. And I know DEC uses it for the Eastern hellbender, which is a very large salamander in slow flowing streams. And it can be very difficult to sample because they're under large rocks on the bottom of these rivers that sometimes are muddy, and people have to actually dive and turn over these rocks to be able to find them. And so eDNA has been a really useful tool for sampling waterways to find out if that salamander is
Yeah, that method was very intrusive, right? Turning those rocks over,
yes.
disturbing microhabitats.
Yeah. And that's what I was going to say, that one big benefit that I see is that lots of times it reduces the need to capture and handle animals. And so it reduces stress on the individual organisms. And so that's one of the pluses. And I wanted to say that before I move on to the last question, which is, let's see, where was this? Oh, Adrian asked, what do you see as negatives to eDNA?
Yeah, I saw that question. We talked about the limitations. That's the only thing that I can think of. I'm not sure if that's exactly what she was getting at, or if she has something else she was thinking in terms of negatives.
Pretty much just the limitations and financial, I would imagine financial.
Well, in terms of financial, it's a benefit, so it's not a negative. You know, extracting environmental DNA has allowed you to step back from all these manual intrusive methods, high numbers of people, vehicles and boats, and all these high expensive ways of gathering data or gathering samples. It saves a lot of money.
Are you getting the funding that companies would normally have gotten for manual labor to get this stuff?
Oh, I don't know. That's a good question. Does it affect funding? Is that what you're getting at? If you're not doing all those expensive ways? I think you're going to spend money maybe in other ways, particularly in the lab, refining the techniques.
But it should be a positive that eDNA is decreasing the amount of money spent obtaining samples. So
It
is
is.
that money
It
being
is.
taken away from, are you getting enough funding for eDNA is what I'm asking? Or are they still fettering it out to manual, even though that's
I
not
I
their,
can't answer your question. I don't know anything about the funding sides of things. I can tell you that they save money collecting the stuff, but there's this, trade-off, because you might be then again spending more money in the lab on refining your techniques or using more highly expensive
okay,
techniques of sequencing and sampling, or sequencing and analyzing.
okay, thanks.